On mute.
Hello, good evening, everyone.
Sir.
Good evening, sir.
Good evening, sir.
Good evening, sir.
Good evening, everyone. Just checking to make sure I'm audible. Krishna here.
Yes,
You are audible.
Yes,
You are audible. You are audible.
Thank you.
We also had some problem with the audibility.
How many participants are there, Ashish?
Sir, around 30.
Yes, good. So do we wait for some more time or?
Another 5 minutes or so. What do you think, everyone?
6:05 we can start. Another 3 more minutes are there.
तू जरा मागे सरक रेश्मा, दिसत नाहीये. चेहरा अर्धवटच दिसतोय.
Galvina is there?
Yes, she's there.
Yes, sir, I'm there.
Yeah, okay.
Yeah.
I was just checking whether the things which I have sent you have read it or not.
Yes.
I think Mr. Sagar Patel is also here.
Should we start now,
Yeah.
Dr. Alka?
Yeah, I think we can begin now. I think we can begin. Maybe BMK sir, if he'd asked us to wait. So if he says we'll start.
I am online.
We'll begin, sir.
Yeah, I think so.
So good evening, everybody. To the Dr. H.P. Tipnis Memorial Lecture Series. I'm saying what would have been the fifth one because due to the pandemic situation last year, we could not hold a memorial lecture. This memorial lecture series was initiated in the year 2017. A year after the death of Dr. Tipnis by the Dr. H.P. Tipnis Endowment Trust. This Dr. H.P. Endowment body, formed by Dr. Tipnis students and wishes from academia and industry. The aim of this body is to promulgate and propagate his thoughts and his rich legacy the future generations and the generations. Only to say a few words.
Alka?
How
Dr. Alka? I think your voice is breaking up.
Yeah.
I don't know whether others are seeing this. The voice is breaking up.
Pardon?
And I request all others to keep it mute except for Dr. Mukune. Otherwise, we have disturbance in the background. Thank you.
No, I've just
Go ahead,
muted
Dr.
everybody
Mukune.
because there were a few mics on. I just muted. So, am I audible?
Yeah, you are now. You are now clear.
Now you're better audible.
Yeah, now you are audible, but in the beginning we couldn't hear what you were saying.
Do you
Sorry.
want me to repeat what I said?
Yes, yes, because your voice was cracking a lot. Couldn't understand anything.
Fine.
Thank you.
Fine. I'll do that. I'll do that.
Thank you, Dr.
So, yeah, thank you, Dr. Reshma. So, good evening, everybody. We have assembled here today for what would have been the fifth memorial lecture of the Dr. H.P. Tipnis Memorial Lecture Series. I'm saying what would have been fifth because last year, due to the pandemic situation, we could not hold a lecture. This lecture 2007, a year after the sad demise of Dr. H.P. Tipnis. And this was done by the Dr. H.P. Tipnis Endowment Trust, a charitable body formed by Dr. Tipnis' students and well-wishers from academia and industry. The aim of this charitable trust is to promulgate and propagate Dr. Tipnis' thoughts and further his rich legacy for the future generations ahead. Now, Dr. Tipnis really needs no introduction, but to say something about him, especially for the younger members from the staff, faculty, and students. Dr. Tipnis has been principal of Bombay College of Pharmacy from 1971 to 95, when he then retired from service. During the from the fledgling institute that it was into the world-class pharmacy institute that we know of it as today. In fact, it would not be an exaggeration to say that whatever awards and accolades college is drawing today, be it the fabulous NIRF rankings or the AICTCI Industry Best Linked Awards, all of these have been possible due to the extremely strong foundation that Tipnis laid out for this college. Tipnis sir has also been very active in the professional sphere through Indian Pharmaceutical Association. He's received numerous awards and honors for his distinctive work. Notable amongst these include the Indian Pharmaceutical Association Raman Bhai Patel Foundation Lifetime Achievement Award, and many more. An astute administrator, a visionary, and a statesman, Dr. Tipnis' legion of students today are literally the who's who of the pharma industry all across the globe. In fact, these 4-5 memorial lecture series, lectures that we've had have been addressed by Dr. Tipnis' students. They have participated, keynote speakers, and really conveyed their tribute to the doyen. Now, today, 7th July, also happens to be Dr. Tipnis' birth anniversary. And on the occasion of this 5th memorial, a 4th memorial lecture, not 5th, of the Dr. H.P. Tipnis Memorial Series, we are fortunate to have with us Dr. Anil Khan as the keynote speaker for the function today. We also have a host of distinguished invitees and guests, Dr. Tipnis' students, associates, and so many more who've joined us here for this function. So a warm welcome to all of you. And with this very brief prologue to the entire event, I now invite Dr. V.M. Kulkarni, formally welcome gathering. Kulkarni has retired as Professor and Dean from ICT, the erstwhile UDCT, and is also the very first PhD student of Dr. H.P. Tipnis. So, Dr. V.M. Kulkarni,
Yeah.
please take over from here.
Yeah. Thank you, Dr. Alka. Am I audible, please?
Yes,
Am I
sir.
audible?
Yes, sir.
Yes, sir. Yes, sir. You are audible, sir.
Okay. Thank you. First of all, I consider this as an auspicious day to the blessing of my Guru, Dr. H.B. Tipnis. Myself keeping very fit at 78, feel pleasure to welcome distinguished people. Good evening first to all those who have joined from India, and good morning to all those who have joined from USA and Canada. Particularly Dr. Khan, good morning. I have a great pleasure in welcoming firstly Dr. Anil Khan, who graciously and immediately on just a WhatsApp message from Dr. Ashish accepted our invitation to deliver a memorial lecture in honor of our sir, later Dr. H.B. Tipnis. He is going to be introduced by the next speaker. This is the 5th event. The first one, as she told, was held in 2017, and very distinguished speakers accepted our invitation, namely Dr. Gopal Kumar Nair, Ajit Singh, and then Dr. Anil Saxena, later Dr. G.D. Yadav, then Director, Vice Chancellor of ICT, later Dr. Professor M.M. Sharma, then Director of ICT, who delivered their very interesting and fruitful lecture related to academic and pharmaceutical research and teaching. With this, just a brief introduction about my sir, which I remember a lot about his teaching and research. One word I like to put it here, that he molded me. Otherwise, I would not have been what I headed the ICTA, still working as a professor emeritus in Pune College of Pharmacy. His blessings have kept me healthy. With this, I welcome Dr. Anil Khan once again to this function. And next, Dr. Mr. Nitin Mania, Chairman, Governing Body, Bombay College of Pharmacy, who is always with us and he has never missed any function of Dr. H.P. Tipnis. We are all grateful to Dr. Nitin Maniar. Welcome, sir. Similarly, Dr. Krishna Iyer, the faculty and in charge principal, present in charge principal of Bombay College of Pharmacy, who is well known to all of you. Welcome, Dr. Iyer. Then Dr. N. Sivaprasad, presently President IPA MSB, who is also always present for this function. Had this function been physically instead of virtual, the interest and the glory of the function would have been very different. But because of this ugly corona pandemic, last year and continuing this year, and new variants are coming, which has crippled the education research activities. Let me tell you a little bit. I am held up in Bangalore last year, 9 months, this year, 4 months. No learning, no teaching. Really, I feel very, very sad that at this moment, we are not able to conduct such auspicious events. But online, physically, it would have been a great, interesting. I welcome once again all the students of Dr. Tipnis, well-wishers of Dr. Tipnis, invitees and guests, and office bearers of Bombay College of Pharmacy, and other members, and also the people from, or the attendees from America and Canada. With these few words, without taking much time, let me tell you, there is no power in Bangalore from morning because of heavy rain. Transformer is burst. Whatever it is a great pleasure that we are conducting this interesting memorial lecture in honor of our late Dr. H.P. Tipnis. Thank you so much.
Thank you, Dr. V.M. Kulkarni, for this warm welcome. I now invite Ashish Bhaktiwale, one of the trustees of the Dr. H.P. Tipnis Memorial Endowment Trust, to give a brief about the trust to the gathering today. Ashish.
Thank you, Dr. Alka. All the respected dignitaries who are attending today to this session, Dr. Kane sir, welcome, and all my teachers. I will not take much time because we are waiting for sir's lecture also. But let me tell you, we still feel that Dr. Tipnis is still around, and I'm not exaggerating, because one thing is that wherever I go, if I tell I'm a Dr. Tipnis student, I do get that respect, and I'm proud of that. So it is a borrowed respect which we also get because of him. And I have been going to Bombay College of Pharmacy quite often last 2 years, and during this lockdown also. And let me honestly share with you, whomsoever I talk, there's hardly any day which goes if somebody has not talked about Dr. Tipnis in the college. So that much the college is linked with him, and that's why our whole focus was with BCP when we started this endowment trust. In the honor of our respected and beloved sir, Dr. H.P. Tipnis, we are having this first time virtual memorial lecture series. Otherwise, we used to have a physical lecture series. So I would just share with you what we have done till date. And then Dr. H.P. Tipnis Endowment Trust has been basically, the purpose was to give study grants to the students, to give academic support to those libraries or inputs or academic finances to the students, conduct the memorial lecture series. We were also planning full day workshop, which we have not yet done. Then skill development program, yes, we are encouraging students for that. Career development, we are just into through some other platforms, we are trying to help the students. We have a board of trustees here. Madam Vinodini Tipnis is there, who is the chairman. Dr. Reshma Tipnis is board of trustee. Ashish, myself is there. And Mr. Prashant Karkanis. We formed this trust in November 2017 as Dr. Alka had shared with us in her session. We have advisory committee, Chairman Dr. V.M. Kulkarni and Vice Chairman Dr. Ratnakar is also there. In endowment committee, under the trust, we have a couple of committees. I'm just going to mention about 2 committees. So an endowment committee where we decide what programs we have to take. Chairman is Dr. Vinodini Tipnis and Vice Chairman Dr. V.M. Kulkarni. The members are Dr. Suman Mahajan, Dr. Mangal Nagar Senkar, Dr. Urmila Joshi, Dr. Alka Mukne, Dr. Jagtap Madam, who is not there now, Dr. S.R. Kulkarni, Pramod Patwardhan, and Pawan Bhatt. The administrative committee is Mrs. Vinodini Tipnis, Dr. Ratnakar. Then the treasurer was late Dr. Pradeep Naik. Yet we have to nominate the vacancy again. And members are Dr. Reshma Tipnis and Mr. Ashish Baptiwali. So this is the administrative committee. So these 2 committees are basically involved into planning and taking decision about the trust. As I shared with you, these are the purpose. And therefore, the first session which we started and the first lecture series which we started was in the 7th July 2017, exactly 4 years ago. And that is the time when Dr. Gopakumar Nair was there. And we were also having a session from Dr. Naveen Saxena. So he was our special invited during that session. The second lecture series was on 7 July 2018, which was physically again held up at Bombay College of Pharmacy, if you can see. And there was a faculty and there was an excellent speech from Padma Shri, Professor Shri G.D. Yadav. And our special guest of honor was Mr. Ajit Singh, who also brought a dynamic addition to the session. We had August audience like Dr. Subhat Priyolkar also there, and you can see the whole auditorium which was full. We had one more session. I don't have the visuals of that. That's why I'm just stopping here. Is that we had Dr. Anantanarayan also coming there and speaking for us. Then apart from that, we had initiating the enrichment of libraries through gifting valuable academic books to the libraries to Bombay College of Pharmacy. We also had a recognition to the teachers who contribute for the image of BCP and also take BCP's name to various sites. So award and recognition to Dr. Mrs. Mala Menon for her service over the year in field of pharmacy and especially being awarded as best professor in pharmaceutics by the National Education Awards. Similarly, award and recognition to Dr. Mrs. Alka Mukne for her service over the year in the field of pharmacy and her paper being awarded a 3rd rank nationally titled Assessment of Chemical Stability of Constituents in Thiosulfonate derived rich extract of garlic validated by HPLC method. We wanted to conduct last year also in 2019 when a couple of our teachers had contributed and they were nominated as best scientist or best awards we have got. But unfortunately, we could not do that. So that is still pending from our side. And in 2019, we have also assisted needy and deserving students with partial financial support for college fees. So we sponsored almost around B.Pharm 3 students and M.Pharm 1 student partially. And in 2020, since 2020 was a great pandemic year and it was a very big issue. And we had also, along with other BCP Alumni Association and others, had helped the students. So that was the time when we also wanted to support to a greater extent. So we supported one M.Pharm student, which was beyond 50,000 rupees. So that is what the support which is given by Dr. H.P. Tipnis Endowment Trust. These are the few activities which I wanted to share with you till now, which the trust has done. We seek your support in consultation for improvement in the activities, also which scientific and information programs we have to do, and some raising financial supports for this. So this is what I just wanted to share before we start the session. So that's all from my side. I will hand over the session to Dr. Alka. Thank you very much for coming, and let us see and enjoy the session of Dr. Anil Khan, my teacher who taught me also. Thank you very much.
Thank you, Ashish, for that brief and but very comprehensive overview of the trust activities that we have done till now. I now invite Mr. Nitin Manial, Chairman of Bombay College of Pharmacy, to address the gathering here. Mr. Manial.
Yeah, thank you, Alka. Good evening to one and all, respected dignitaries, madam and dear friends. At the outset, I would like to express my gratitude towards Dr. H.P. Tipnis for his continuous support, mentoring, and guidance since my association with him of the last 2 decades. I'm not the only lucky one, but n number of the professionals who had worked under him and got nurtured and now serving as a renowned professionals. Friends, we debate sometimes. Sanstha Mahan ki Vyakti Mahan. Of course, Sanstha. But the Sanstha is made by the Vyaktis only. Likewise, BCP was earlier recognized by Dr. Tipnis College. Yes, it was Dr. Tipnis College. Let me tell you, he was very much possessive about the BCP and the IPMSB. He never allowed anyone dissociating the BCP and the IPMSB and remained on the forefront whenever any issue had arisen. He had been witness for both the glorious period of the BCP, that is the silver and the golden. He was a bridge in between, and during the silver period, exclusive development of the academic excellence followed by the golden period of the research, and thus making it the academia plus research that comes to industry institute partnership culture, and that he nurtured well in the college. Today, whatever the fruits that we are enjoying at the BCP, that is name, fame, infrastructure, compliance, exemptions, expansions, etc., majority has taken place during the era of the Dr. Tipnis only. Friends, there are not only words or lip songs, but it's proven. It might be the income tax exemption under 35(1)(2), establishing the Society for the Pharmaceutical Industry Research, Amrut Modi Research Fund, Research Society Bombay College of Pharmacy, Dr. M.K. Ranganekar Memorial Testing Laboratory, Auditorium, Staff Quarters, Pacific Wing, CSIR approval, and many more that has been occurred during his tenure only. He was the main pillar as well as the mentor for the IPMSB, and not only conducted the free and fair elections of the IPMSB since 2004 till 2016-18, and almost 7 elections, and not only conducted the IPMSB, but also the local branches that include the Pune and the Nagpur also. I would like to share a small incidents like how meticulous he was. When he shifted from Powai to during the redevelopment of his old building, and afterward he returned back to the original residence, which is at Ville Parle. But for this period also, he had informed to IPS Center and to the IPMSB also in the writing for the change of the address on the temporary basis. And again, after the shifting to Ville Parle, and that shows how meticulous he was about the documentation, transparency, and in small things. Friends, we sincerely appreciate the concern of the HP Tipnis Endowment Fund, Endowment Trust, to support the BCP and the students of the BCP. We thank you, Madam, and the students of the HP Tipnis for the same. Madam, we assure you our wholehearted support for any such move, which would be further enhancing and strengthening the image of the BCP being the premier institute in the country, which is also recognized by the AICTE, CII, NIRF, and NBA in the recent past. Thanks for inviting us for the today's lecture and giving me an opportunity to cherish some of the memories of the sir. Thank you very much.
Thank you, Chairman Sir, for giving us the overview of BCP plan and past activities and future plans, and sharing your valuable experience that you had with Dr. Tipnis. So thank you very much. Now I take this opportunity to invite our in charge principal, Dr. Krishna Iyer, to speak to the gathering here today. Dr. Iyer.
Good evening, everyone. And I presume I'm heard. My microphone is working. Hello.
We are able to hear you.
Yeah. Good evening, everyone. And it's a great honor for me to be part of this evening and being part of the memorial lecture for Dr. H.P. Tipnis. I have been Dr. Tipnis' student when I was in B.Pharm from 82 to 85, and I've seen up close and personal how amazing Dr. Tipnis was, both on the academic front and on the administrative front. And therefore, it is with a little bit of nervousness that I'm now doing this job of in-charge principal, a task which is not very easy. And such an amazing institute put up by such an amazing person has been given to you to kind of maintain it for a while as in-charge principal. It comes with a little bit of nervousness. Okay. But I would like to take this opportunity to pay my respects to Dr. Tipnis, to all my teachers who are there in the audience today, and my regards to all the peers and colleagues who have been part of BCP when I was a student. Like, for example, Dr. Kane, who also has taught me, Dr. Ratnakar, Dr. Baptiwale, and so on. Okay. It is great to be part of this, and I'm very happy that I have this opportunity to be part of this in a different light rather than simply an attendee. Okay. And I wish the endowment all the very best. And if any assistance is needed from my side, please do call upon me, and it will be with absolutely no issues from my side. Good evening, everyone, and I look forward to the talk of Dr. Kane. And regards, Dr. Kane, and welcome. And welcome again to all the people who are attending. And thank you, Dr. Reshma, and my respects to Dr. Tipnis's wife, who I have not had the opportunity to meet personally. I would love to do so at some point. Thank you very, very much. And Dr. Alka, please.
Thank you, Dr. Iyer, for your address today. I now invite Dr. N. Shiv Prasad, President IPA Maharashtra State Branch, to share his thoughts with us today. Dr. Shiv Prasad.
Thank you, Dr. Alka. Very good evening to all of you. Of course, good morning to some of you are from US and Canada, as it was told by Dr. Kulkarni. And thank you also for inviting me to this evening lecture in honor of Dr. Tipnis, who was a great teacher and educationalist. And I also take this opportunity to welcome you all on behalf of IPMSB, Indian Pharmaceutical Association, Maharashtra State Branch. Though Dr. Tipnis is not physically present here, he lives in our memory and mind. And he has tremendously contributed to the pharmacy education, particularly when it was in the infancy state. And my association with him was very long. I must say it was long and deep, because in the year 1985 or so, 84, 83 time, I first met him. It was he and Dr. Balla. Because of them, I have the long association with the Bombay College of Pharmacy. I must say, long, in-depth relationship with the Bombay College of Pharmacy that is still continuing. In fact, my first lecture on statistics was arranged by Dr. Tipnis. At that time, it was not a subject, but he wanted to have this subject for their research student. I still remember at that time, Dr. Mala Menon also attended my lecture. And I told that he is a great teacher. It's not because that he was having a of course, he had a great skill in teaching, and he had got an in-depth knowledge in his subject that he taught. But he has a passion for pharmacy education. Pharmacy teaching, he had a great passion, and he has also had a passion for the student to acquire adequate, clear knowledge and skill. And also he had a great passion for the Bombay College of Pharmacy. He wanted to make it a fine academic institute that you know that it is one of the fine academic institute. As Mr. Maniar explained, it is all his effort, and he has put effort in many areas, particularly the network of industry and industrial collaboration. And in fact, I must say a research culture he has developed. He used to say that research should go hand in hand with the academic activities. That will strengthen the academic activity, that in turn strengthen the research. Now it is our faculties are all having that mindset, and they are all involved in the continuous good quality research. And there are many awards and recognition which are testimony to this research culture. And then I also understand that the Research Society of Bombay College of Pharmacy, which is created by Dr. Tipnis, is his brainchild. And it is a very unique concept, because this research will be managed by the Research Society of Bombay College of Pharmacy, but it will be executed by its faculty. So there is a link. So this will enhance the effectiveness in the research area. As Mr. Maniar explained here, he was very much associated with the Indian Pharmaceutical Association Maharashtra State Branch. I must say that he was a friend, philosopher, and guide. Why I used the philosopher particularly, he had a clear thinking. He is a very great thinker, and he takes decisions or suggests solutions which are very practical, and it is a wholesome. It is a holistic view it will have. So in later period, we will find that its benefits and results in a better way. And another thing, important quality I have seen in him is wherever he involved, he take it very seriously, and there will be an active participation, and he will see that the results of that involvement or the participation results in a good return. I will narrate one experience. I had several experiences with him, but one of them, this I would like to suggest. Myself and Dr. Tipnis were invited by a pharmacy college to give 2 lectures, one workshop with 2 lectures. He was to talk on how to write research papers for the students. I was to give a talk on how we can use statistics to make the research paper more attractive. Though his subject was separately, he was invited, I was separately invited, but he came to know who is the other speaker, and he telephoned to me and told me that, see, I know the pulses of students. So I would like you to emphasize certain points. And he made a list of points and sent it to me. And thereafter, we went both for the lectures, and he gave his lecture first, and then I gave a second. He was to go somewhere else from there. It was, I remember, it is an Igatpuri. From there, he was to go somewhere, but he stayed back and attended my lecture. At the end of the lecture, instead of others, he asked several questions, but which I know that he knows that answer, but he has asked. So later on, I have, after came to Bombay, I asked him, you have asked several questions for which answer are known to you. So he told that the students are very shy to ask questions. They will not ask. To make your lecture more effective and more clear to the student, I myself asked a few questions. So he was, that means he participated, not just participated for the namesake. He has actively participated. The involvement is very great, and he aimed at the good result out of this. That was Dr. Tipnis. So I enjoyed his association, and making the Bombay College of Pharmacy still better institute for academic and research will be the greatest tribute, according to me, to Dr. Tipnis. And thank you once again, and I wish this program all success. Thank you.
Thank you, Dr. Shiv Prasad, for sharing those wonderful anecdotes about Dr. Tipnis. After Dr. Tipnis' passing away, Dr. Reshma Tipnis and Mrs. Vinodini Tipnis have sort of been the fulcrum and the central pivot around which sir students from all across the globe have come together and formed this trust. Now I invite Dr. Reshma Tipnis to speak to us about the plans for this trust. Dr. Reshma Tipnis.
Thank you, Dr. Alka. Good evening, everyone, and good morning, Dr. Kane. Good morning, USA and Canada. Respected dignitaries, all students and well-wishers of Dr. Tipnis. I'm very glad to see all of you attending this session. Thank you so much. Last year, due to COVID-19 pandemic, we couldn't conduct this memorial lecture, and we were hoping that one day we'll be able to do it physically at Bombay College of Pharmacy, as we have always done it before. But we just couldn't do that. So this year we decided to do it virtually. Basically, we didn't want to miss this date, 7th July, as all of you are aware that it used to be the birthday of my father, and hence this Dr. H.P. Tipnis Memorial Lecture is held every year on this 7th day, on this particular day. Today, I would like to share the recent development of Dr. H.P. Tipnis Endowment Trust. Due to lack of manpower, logistic reasons, and some other issues which we didn't foresee, we have decided to hand over the trust so that the mission and the vision of Dr. H.P. Tipnis will continue ahead. We are already in discussion Bombay College of Pharmacy, IPA, MSB for the same, so that they will be able to do many more activities, much more than we could do in last few years. And the objective of the trust, which is for which the trust is made, that will continue forever. So hoping for fruitful closure of this trust soon. And I know all of you and myself as well very keen to listen to Dr. Kane now. So thank you so much, and I'll hand over to Dr. Alka. Thank you.
Thank you, Dr. Reshma Tipnis. And now I invite Dr. Ratnakar Mahendra, one of Sir's very loved favorite students and a very active member in the trust, to introduce our keynote speaker today, Dr. Anil Khan. Over to you, Dr. Ratnakar.
Yeah, thank you, Dr. Alka. One advantage I should say about virtual meeting is that we can have an international speaker speaking at any given time according to the convenience of the speaker. And when I'm looking at the participants, I can see Dr. Darshana Hegde from Shanghai. I hope she is in Shanghai. And I can see Dr. Ashok Ghone from US. So at this moment, I can have any person from any part of the world attending the session and gracing the session, because it's a session in the memory of our beloved Dr. H.P. Tipnis, and we are so indebted to him, all of us. So it's an honor and a great pleasure to introduce my senior and my teacher, Dr. Anil Khan. Dr. Anil Khan is the global head of Technical and Scientific Affairs, Tatian Incorporation, which is part of Thermo Fisher Scientific. He has more than 25 years of experience in the science and business of taking molecules through the entire drug development process. His extensive knowledge spans early stage development to scale up and commercial manufacturing, and includes technical transfers between global sites and drug lifecycle management. Dr. Kane received his bachelor's, master's and PhD degrees from the prestigious Bombay College of Pharmacy, University of Bombay, India, and served as a postdoctoral fellow at the School of Pharmacy, University of Cincinnati, Ohio. He has also earned an executive MBA from Richard Ivey School of Business, University of Western Ontario, Canada. Dr. Kane is a member of various international pharmaceutical professional organizations and is often asked to speak about scientific topics on formulation, technology, and other technical aspects, etc., at major industry events. And I remember meeting Dr. Kane in US and in many CPHIs in Europe as well. And whenever I meet, I think we have also taken photographs with him. And it's a great pleasure and honor to meet him and many of our seniors in most of the conferences, seminars, and CPHIs internationally. He has also published many articles in international journals and delivered many talks at meetings and conferences across the globe. I profoundly thank you for accepting this invitation. And now I request Dr. Anil Khan to kindly take over us through this presentation on new avenues for growth of pharmaceutical industry in next decade, which is going to be a big challenge in the pandemic. And I hope the pandemic doesn't stay for long. So over to you, Dr. Khan.
Thank you very much, everyone. Thanks for the kind introduction, Dr. Mahendra. Good evening, everyone in India, and good morning to those in US, Canada, and I believe it's good afternoon in Shanghai. So thank you, everyone, for participating here today. First of all, Dr. Alka Mukne, respected Dr. V.M. Kulkarni, Dr. Bapti Wale, Dr. Maniar, Dr. Krishna Iyer, Dr. Siva Prasad, Dr. Reshma Tipnis, Mrs. Tipnis, and the entire committee. Thank you very, very much for inviting me to speak today. It is a privilege and honor to participate in today's program and speak. As you heard from the introduction, I've had the honor and privilege to be a part of Bombay College of Pharmacy for almost 10 years. And as a student, of course, from completing my bachelor's, master's, and PhD from Bombay College of Pharmacy. Hence, my association is for a long time, and I'm indebted to Dr. Tipnis. What can I say more as that has not been said already? Dr. Tipnis was a great academician, researcher, advisor, mentor, a great teacher, an excellent administrator, and of course, beyond all that, a great human being. I was proud and continue, and we all on this session, many of us are students of Dr. Tipnis, and we continue to be fortunate to be his student and to work closely under his guidance at the Bombay College of Pharmacy. Dr. Tipnis was a great visionary and always used to look ahead. I remember him as one example being a pioneer to bringing biopharmaceutics, bioavailability to India, and being a leader in bringing new concepts, new ideas into the pharmaceutical world in India. On that note and that theme of Dr. Tipnis always wanting to look ahead, I have prepared my talk today, and I will now switch to my presentation as to how we can look ahead in the pharmaceutical industry. Just to check if you can see my slides and if I'm audible.
Yes,
we can see your slides, Dr. Kane.
All right. Thanks everyone again for participating. It is again an honor to present today's memorial lecture. I will be speaking about new research, new avenues for the growth of pharma industry in the next decade. Many of the concepts will be new to everyone. They are new to everyone on the globe. And I'm sure and I hope that there are some key takeaways from the information from this talk. If we look at the development of medicinal drugs, traditionally, they have been of synthetic origin as small molecule active pharmaceutical ingredients. They could be as single ingredients or a combination. Or in the last decade or two, we have seen the emergence of large molecules, biologics or biosimilars, such as recombinant proteins, fusion proteins, monoclonal antibodies, and others that are being developed for treatment of a variety of indications, a variety of disease states. And at the bottom of the slide, you will see the traditional drug discovery process for molecules that are coming out of drug discovery, screening them, testing them through various clinical phases from phase 1, 2, 3, and being approved to be used in the market for the treatment of several indications. In the last decade or so, we have seen the emergence of drugs as repurposed drugs, which means for the same drugs are being repurposed for newer indications. That cuts the time of bringing these drugs to the patients and to the market by almost half. Traditionally, it used to take about 10 to 17 years. And with repurposed drugs, it takes about 3 to 8 years. Some of the technological advances that has helped drug repurposing are the fast computerized computational tools that are essential for bringing information from the big data, QSAR modeling, computational tools like machine learning, neural networks, artificial intelligence, etc. These are the approaches that has helped speed up the drug design process and help bring repurposed drugs to fruition, to clinical studies, and to approvals. If we look at what the drug repurposing trend has been, apologies for a very busy slide, but what I wanted to show here that several computational tools and experimental approaches from molecular docking, novel data sources, and signature matching have been used to check and evaluate existing drugs for newer indications. There is a huge market for repurposed drugs. In the last decade or so, we have seen many drugs being repurposed for newer indications. A great example is in the pandemic. We have all seen that drugs that have been used for many other indications have been tried to treat pandemic and other indications as well. On the right-hand side, you'll see some interesting data that it is estimated that 30% of the FDA-approved new drug products and repurposed drugs account for almost a quarter of the pharmaceutical industry revenues. And this is a big number that validates the use of existing drugs for newer indications. These drugs and the new indications that are being sought after are everything from diabetes to oncology, autoimmune conditions, CNS disorders, and cardiovascular disorders as well. So drugs are being repurposed for many other indications. Looking ahead and beyond, and I will be focusing today on the next 10 years or so, what are the trends in research? What are the trends in the pharmaceutical industry in the developed part of the world, along with many other companies in the global field? And this would bring newer medications, newer avenues to the patients and a business opportunity as well. So in the rest of my talk, I will be talking on these topics, probiotics and human microbiome market, oral biologics and biosimilars. What does mRNA vaccines have to offer for not only treating infectious diseases, but other therapies as well? What does cell and gene therapy have to offer? And what does precision medicines have to offer? As you can imagine, these are newer areas of research. However, there are many companies already that have been successful. And here is an opportunity to bring better medications to the patients and business opportunity for the pharmaceutical and healthcare industry as well. Firstly, let's look at probiotics and microbiome. Let's understand some definitions here. What does microbiomes mean? Human microbiomes are microbial communities that live inside our body. Microbiomes contribute to a broader genetic portrait or metagenome of a human. We all, as human beings, depend on a vast army of microbes to stay alive. For the proper balance of these microbial flora in the gut, and this protects us from a variety of infections, germs, but also helps to break down the food and to release energy. These microbiomes provide valuable information regarding human health and disease predisposition. On the right-hand side, you'll see the human gut and several microbial flora that exist. And the balance of this is extremely important. And as one can imagine, no one microbial flora in one human being is similar to the other. It all depends on a variety of factors: the food intake, the lifestyle, the environmental conditions, and a number of other factors as well. And why is this important? This is important to prevent other infections and disease states. Let's look at what probiotics means and why many a times these need to be substituted to the human body to preserve the microbial flora. Probiotics are termed as friendly bacteria because these are consumed to balance the gut flora. Often there are some confusions between what is prebiotics and probiotics. Prebiotics are compounds in food that induce the growth and the activity of the beneficial microorganisms as bacteria and fungi. These prebiotics are a part of the food that we intake, and this preserves the microbial flora as well. Why are all these important here? Substitution or providing these probiotics have many beneficial effects. They are important as immunological activity. They aid in digestion. They protect us from other pathogenic bacteria. They improve lactose intolerance, which is on the rise globally. They decrease cholesterol levels, and more research is being done to understand the impacts of probiotics. And hence, I say that this is a newer area. Many clinical trials are being conducted to test these probiotics in the treatment of Crohn's disease, ulcerative colitis, intestinal bowel syndrome, and many other immunological disorders, including cancers. If we see these probiotic bacteria and what are these bacteria that needs to be substituted, this is the big list of bacteria that have various functions and benefits, as we saw on the earlier slide, and have activity. When we talk about probiotics, the first thought that comes to our mind is that, oh yes, we have heard commercials, we've heard several promotional material by the media to provide these probiotics. And these probiotics have been formulated and provided to us in the form of food supplements: cheese, yogurt, creams, chocolates, milk, meat, etc. However, these non-conventional methods of delivery are sometimes questionable. The viability of these and the survival of these probiotics are not essentially controlled. Today, we have better techniques in the pharmaceutical world to formulate these probiotics in better formulations, in better dosage delivery formats, such as beads, pellets. And since these are live bacteria, as lactobacillus and others, these need to be preserved from the time they are formulated. Their activity needs to be preserved until they are delivered to the right part of the GI tract for them to act in the microbial flora. Hence, there are opportunities to formulate these better. Many a times we see these bacteria as lyophilized bacteria, frozen. Our cryoprotection and better delivery systems like protecting from the gut, enteric coatings, and colonic delivery, etc. So today, with better techniques in drug delivery, pharmacists and formulators can better deliver these probiotics for as better mechanisms for them to have a much better impact, and these could be tested into clinical trials as well. Before jumping into seeing why these are important, there are some terms I'd like to make sure we have an understanding. And these are areas of different studies. So proteomics is the study of proteins. And this is important to understand the molecular biology and understanding the proteins and genomes of an organism. Genomics is nothing but a study of genomes, or understanding the set of genes in a cell or organism. And metabolomics is the study of metabolites present in the cell or tissues. These are important fields of science that are now being researched to have a better understanding of the impact of a number of items that we'll be talking about in today's talk. So back to the human microbiome market. Why is the microbiome is important in research and clinical efficacy? If you see the trend and the forecast, you will see that these probiotics and delivery mechanisms that we talked about seem to be gaining importance. And these are, in the form of drugs, going to be growing. The industry is growing in double digits. So more than 20% are being tested in drugs and others as diagnostic ingredients. If you see this slide, you will see that the human microbiome market is huge. And for therapeutic applications, you will see that it's a big trend, and a lot of research is being done to treat infectious diseases, GI diseases, endocrine metabolic disorders, as well as cancers. See this microbiome market being looked at in different parts of the world. We are seeing a big trend not only in North America, Europe, but also in Asia Pacific and rest of the world. You'll see a big market share growth, and this is going to be an important aspect in the coming years and the decades. This is the delivery system that will play a big role in the healthcare industry. And that's where the understanding of genomics comes into play, understanding of genes, metabolites, protein chemistry, and cell biology is going to play a big role. There are many prominent players already in this industry researching, and you'll see many companies in this area, not only in North America, Europe, but in other parts of the world. And so there is a trend that a lot of investment being done with some companies in India as well. The next topic I'd like to touch upon is oral biologics and biosimilar market. Traditionally, the biologics have been delivered via the injectable route, simply because these biologics are not stable in the acidic pH in the stomach. Because of the acidity, these biologics get inactivated, and hence they are not functioning as they should. A big example is the large molecules, proteins, peptides, and insulin, for example, are provided in injectable route only. But a lot of research is being conducted in delivering these by oral delivery, which of course has benefits. The global oral biologics and biosimilar drug market is expected to grow also in double digits. And you'll see the numbers here on the slide that there is billions of dollars of market in the coming years. This is going to grow more than 20%, not only in North America, Europe, but in the entire world. Delivering biologics and biosimilars by an oral route is explored. One point about delivery mechanisms by the oral route or manufacturing these oral biologics as well as the probiotics is that these being live bacteria, they cannot be manufactured or processed in the same plant from a safety perspective, from a regulatory perspective, and hence they require dedicated, segregated facility from the regulations point of view. That also could be investment intensive and could be restricting many other players entering the market. Oral delivery, as mentioned earlier, have been given by the injectable route, as mentioned earlier. But newer delivery mechanisms, cryoprotection, enteric protection, as well as targeted delivery mechanisms that the pharmaceutical industry have researched, have made possible providing these oral biologics into different parts of the GI tract. Dual targeting as immediate release, controlled release has made this possible. And we see a big trend already. There are many players in the oral biologics and biosimilar market. You will recognize many pharmaceutical giants as well as companies all over the globe, including those from India entering into these markets to develop and bring better therapies from oral biologics and biosimilars to patients, and also a great business opportunity. The next topic I'd like to touch upon is mRNA. I think in this pandemic, beyond the healthcare industry, beyond the medical, pharmaceutical professionals, even the common man has heard about mRNA because of COVID. And there is a huge market, not only in the vaccines area, but beyond as well. And let's see what mRNA has to offer in the healthcare industry. How and why is RNA or messenger RNA an important mechanism of delivery and the activity in our human body? If we go back to the basics of cell biology or molecular biology or biochemistry, we understand that the DNA is a storage function in the nucleus for proteins. These DNAs can be transcribed into mRNA, and these mRNAs or messenger RNAs act as a software, and these can be delivered or translated into the proteins as delivery mechanisms. And these can be coded, a proper sequencing or a code can be sent via these mRNAs into these proteins to develop and act the way we want them to act. And that's an example of how this genetic coding has helped to develop protein against the COVID virus. If we look at the mRNA demand, even prior to the COVID vaccine, it was estimated that the mRNA market would grow by a very big folds. If you see the steep growth curve of the mRNA, you'll see that it was expected to grow. And now with the importance that mRNA has played in the two most widely used vaccines from Pfizer as well as Moderna, they will be very important player in vaccines and beyond as well. At the bottom of the slide, you will see that these mRNA are being used in many, many indications beyond the vaccines. There are hundreds of mRNA drugs in development. Most of them are in the preclinical stage. Many of them are being studied for the coronavirus, but not only for the COVID, but there are many other non-COVID indications for which they are being evaluated in the phase 1, phase 2, as well as phase 3 clinical trials as well. Another source shows that there are over 100 or about 120 clinical trials using mRNA drugs. Many of them, of course, as one can imagine, in phase 1. And there are many advanced or late stage clinical trials using these mRNA compounds. An important area of application of mRNA we see is not only for infectious diseases, as seen from the top of this chart, but that to treat various virus infections or viral infections, but also many oncology indications as well. And both these are coming from sending the genetic code into the blood cells to create immunity. And this immunity will fight not only the virus, but it can fight other cancerous indications as well. And you'll see these mRNA medications are being tried in a variety of different cancer indications. There are many.
Sir, excuse me. I am sorry to interrupt in between. I don't know about others, but couple of us are only at the probiotics delivery mechanism slide.
Oh, apologies for that.
No, it's not moving. Not it must be some system error. So not. I thought for the benefit of all, I can interrupt. Thank you very much, sir.
Ashish, I think if you leave and join again, the problem could be sorted out.
I could see till now properly.
Yeah. Can you?
Yeah, we can see now. See?
All right. Okay.
Thank you, sir.
Yes.
Apologies if this was anything from my end. All right, so let me continue in the spirit of time. So many companies are already into, testing mRNAs and researching and bringing these medications to the patients after clinical trials and approvals, of course. Many companies may be familiar to many of us in the pharmaceutical and healthcare industry. And many of these programs, as seen from this slide, are in various different phases of the clinical studies. If we move to the next slide, first application of these mRNAs are, of course, in the current pandemic, very important application that is being done is vaccines. We know that several vaccines are already approved using mRNA. An interesting number to note at the top of the slide, you will see that around 90 plus vaccines are already in clinical development for COVID. We know that only a couple or a handful of these are being approved, but as you can imagine, those ahead of the game have had the benefit of bringing treatment to the patients as well as business opportunity as well. The point I'd like to point out that these mRNAs have a great application for treating infections. If you see this slide, there are a number of infectious diseases on the left-hand side, from malaria to tuberculosis, polio, etc., where vaccines have been developed. Of course, these have been developed by the traditional route. And we'll come to the mRNA in a bit. The COVID vaccine from Pfizer, BioNTech, as well as the Moderna have been the first mRNA applications in the form of vaccines. The chart shown on this slide shows the number of years it has taken to develop a vaccine, and you'll
for two.
Good evening, all. I think there is some technical error, and just for a couple of seconds, Dr. Kane will join back. Sorry for this inconvenience caused. Thank you.
ते पण जस्ट विचारून घ्या की कडे असेल तर ठीक आहे, असं तर त्यांना सांगायला लागेल की हे तू आम्हाला दे, म्हणजे आम्हाला हे बनवून त्यांना द्यायला लागेल.
मागच्या वेळेला का ते आलेले होते ना त्या दिवशी, तेव्हा ते म्हणाले की अग्निवेशने सगळं बनवलेलं आहे, फक्त काय 1-2 डॉक्युमेंट्स नाही.
I'm trying to reach him, but could not reach him. Anybody in the USA can try that?
Apologies, I'm back. I'm not.
Welcome, sir.
Apologies.
Thank you. You are back. Welcome, sir.
Welcome, Dr. Kani. Welcome back.
Yeah, apologies. I'm not sure where I lost you, but maybe I can continue if I can be heard okay now. Can you hear me okay?
We can hear you. We can see the slide.
Okay, I will switch to the presentation mode and continue. All right, so again, I'm not sure where I had left off. Can you see my slide?
Yeah, maybe you can go back a few slides. That's where we lost it. Mr. Kane?
Yes, let me go back a few slides. Bear with me for a second. Is that good?
A couple more back.
Is that visible now?
Yeah, it's visible, but you need to go back a couple more slides.
One minute. mRNA wala.
Yeah. Vaccine innovation.
Yeah, here. It was at the end of the slide.
Yeah, I think the next slide probably.
Yeah, just about here.
Yes.
Yes.
Okay, apologies for that connectivity issue. All right, so as you know, we can see on the top of the slide, there are more than 90 vaccines being tried for the COVID. There are some front runners, of course, but many are behind being studied in various phases of the clinical studies. If we see the slide, there are vaccines developed traditionally in the last 100 years for a variety of infections from malaria, tuberculosis, polio, typhoid fever, etc. And the bars show the number of years it has taken to develop these vaccines. And many of them have taken tens of years to come up with a vaccine. The exception being the last one, the mRNA-based COVID-19 vaccine, which was brought to the patients in the same year as being tried in the clinical studies. So the speed at which the new delivery mechanisms, the new understanding of the mRNA shows promise is to bring vaccines as required very quickly after a limited clinical testing to the human patients to treat certain infections. Some basic requirements and challenges of the vaccines. An ideal vaccine should show the following properties. They have to be safe and efficacious, of course. In clinical trials, they need to have a lasting immune response. They need to be easy to administer. And they have to be available in large quantities. They should be, manufacturable at a very large scale to be able to provide to millions and billions of patients worldwide as required, for example, in the current pandemic. Of course, they have to be economically accessible, thanks to all the pharmaceutical industry, the governments as well, for the making these available. But this is an important aspect of vaccines and an important requirement as well. From a formulation perspective, these vaccines need to be simple and should not necessarily need a lot of adjuvants, diluents, and would need minimal manipulation by the nurses. If we look at the traditional platforms that have been used for vaccines, there are live attenuated viruses, inactivated viruses, and virus-like particles that have been used in traditional technology to trigger the immune response from the cells in the human body. The next generation of platforms now uses viral vectors, which could be replicating or non-replicating. There could be DNA-based vaccines such as adenovirus, lentivirus, and other DNA-based, or the mRNA-based viruses, vaccines, example being the mRNA vaccine made by Pfizer, BioNTech, Moderna as well. Or one could present antigen-presenting cells for developing these vaccines. Some pros and cons of traditional technologies for vaccines. The pros are that these are already present in the market. There is a strong and robust historical data and a very good understanding of what needs to be done from evaluating the safety and efficacy of these traditional platforms. The downside being that they really need to be tested for a long time in a huge population for their safety and side effects as well. Demonstrating that virus will not regain the virulence is extremely important, especially when the live virus or attenuated viruses are being used to develop these vaccines. As against newer platforms, for example, the RNA-based or DNA vaccines, these can be developed using the new understanding of genetic sequencing codes. And the good part is that safety is not an issue here because these are synthetically prepared in the lab in a test tube to begin with and then scaled up. They do not involve any culture or fermentation, and they are relatively easier because they do not have a live or attenuated virus in them. The downside or some disadvantages of the new generation platforms is that they do not have a history, and these are new, as we all know that these are areas where they have not been proven. The first mRNA vaccines to be in the industry was the Pfizer, BioNTech, and Moderna. I think we have lost the connectivity again. Can you see my slides?
We can see your slide now.
Yes.
Okay. All right. So there is a huge market is what I wanted to communicate here, not only for infectious diseases, but for many other indications beyond epidemics, pandemics, and infectious diseases as well. So vaccines are here to stay, hopefully not for any more pandemics, but for other indications such as cancers, where they have shown promise and will be able to treat different types of cancers, hopefully relatively easier mechanisms of delivery and a better treatment platform. One of the quote on the vaccines was, how can we be better prepared to face pandemics if they come our way in the future? While it is not possible to prepare a vaccine for every virus ahead of time, we certainly can leverage our detailed understanding of how to develop a vaccine, how to treat it, and better take care of the human population on the globe by rapidly testing and delivering these vaccines in the future. I wanted to quickly touch upon a few other aspects of research and growth is cell and gene therapy, and just quickly going through what this has to offer. Cell therapy and gene therapy, the definitions on the left and right for our sake, cell therapy is the treatment of a disease by using our own living cells to replace damaged or diseased cells or tissues to stimulate immune response or regeneration. And these cells can be modified and treated by receiving these cells from a donor cells, and these could trigger the immune response required to treat a variety of different indications. Gene therapy is a treatment of a disease by replacing, altering, or supplementing a gene, understanding which gene is absent or abnormal, and how these genes can be replenished, and how these genes can be modified in the cell and delivered as a therapeutic agent to trigger the immune response in vitro or in vivo. And this has been researched in the last several years, more so in the last 5 years than in the decade. And hence, this is going to stay for the next little while in the next decade for sure. We'll see some examples moving forward. At the bottom of the slide, you'll see some pros and cons or the comparison between cell therapy and gene therapy as well. If we see some examples of FDA-approved gene therapies, examples come to our mind are Kymriah, Yescarta, Luxturna by various companies, and a lot of research is being done. And this is the proof that cell and gene therapy have shown promise, and not only in research, but also have brought effective therapies to treat a variety of different indications. I think I've lost the slide again. I will reconnect in a second here. Can someone confirm you can see the slide?
We can see the slide.
Yes, we can see the
slide.
All right. So the cell therapy again has a huge market. How about now? Can you still see the slide?
Yes, definitely.
Yes, we can.
Okay, for some strange reason, the view on my end has disappeared. So apologies.
It is gone off now. We could see it when you asked, but now we are not able to see.
Okay, give me a second and I'll be back. All right, I hope you can see now.
We can see it now.
Okay. Please jump in as soon as you see that it's frozen or disappeared. Sometimes it's very difficult for me to see if it is disconnected.
Sure.
All right, so I'll speed up for the sake of time. We'll see a huge growth in cell and gene therapy market. As you can see, that this cell and gene treatment can be helpful in a variety of different indications, mostly in treatment of different types of cancers. Because this is related to immunotherapy and immuno-oncology. We will see a huge market and big growth in double digits for the cell and gene therapy market. A variety of different vectors have been used in gene therapy. These are from lentivirus, retrovirus, modified simplex viruses, etc. And this is another science and a field that is growing in understanding demand, and there is a huge interest in the global pharmaceutical industry. Let me touch upon slightly, very quickly, of course, in precision medicine, which is gaining importance. What is precision medicine and why is it emerging? The precision medicine is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, lifestyle for each person. Now, this is important because in the newer techniques, in the newer methodologies of understanding individuals and variability of genes and environment, one can cater to titrating the needs to treat indications. So in precision medicine, we can understand the patient's history, the vital signs, electronic health records, and the faster triggers of patient testing and results. Look at all the understanding of the genes, the transcriptions, the protein chemistry, and fast tests that can send signals to understand the titrating the medications as well. And a good understanding of the patient's lifestyle helps to titrate these medications to an individualized treatment. Why is this being looked at? So on the right-hand side, we see that today's drugs that are being developed and being used for the treatment, there is data to show that most available medicines don't work for everyone. Why do we say this? Is that a certain dosage of medicines is being used to treat millions and billions of patients, and this is not entirely accurate because different individuals require different doses of drugs to treat different ailments. And we see that about only 57% of the patient population respond to diabetic drugs, arthritic drugs, Alzheimer drugs, and far less cancerous drugs. What this means is that the side effects of these drugs, while are predominant, and let's take an example of cancers. There are healthy cells that are being impacted by these medications because these are not necessarily targeted deliveries. So by precision medicine, what is being looked at and what is being developed is how can we target these drugs better to have a better treatment, a much higher response to these types of treatments. And this precision medicine is going to be another field of science that will be holding promise for the future. And, again, a new area only coming up in the last 5 years or so. There is a big history, but a lot of investment and research being done in the precision medicine market. As you can see, this will be growing in the next 10 years or so. And this has a billions of market, not only in the North American, Western world, but also in Asia Pacific and the growing emerging markets as well. And this will bring better treatment to the patients and better business opportunities to the pharmaceutical industry. The key drivers for this precision medicine are the increasing awareness, better technology, better understanding of the genes, the proteins, the metabolism, and better scientific pool, better skill sets, and better scientists across the globe that can bring better medications to the patients in the coming decades. So why are we talking about all these newer things and what are the opportunities? I like some of these sayings here. If you want different results than what you're getting, we have to try different approaches. We have to jump into newer areas of research. We have to invest time, money, efforts, and skills to do something different to get different approaches. If we want to be ahead of the game, we have to look beyond and take the plunge. And looking into new sciences, new areas of research, there's always a high risk. But embracing high risk is also going to be rewarded. And these are some of the examples and areas of research that will give us better medications to the patients and better opportunities for the pharmaceutical industry. I will conclude the talk today by concluding in a few key areas. We have seen new areas of research and growth in the pharmaceutical industry in probiotics, oral biologics, mRNAs for cancers and vaccines are being developed. We have seen some cell and gene therapies that are going to show promise. Precision medicine, digital science, and personalized medicines are being explored and will see better treatments for the patients. I would summarize that investment in newer technologies and building the infrastructure and investing in key areas of growth will hold a better future for all of us as pharmaceutical scientists and healthcare professionals. Again, thank you very much for your patience, your time, your understanding, and it has been an honor and privilege to deliver this memorial talk. Thank you again to everyone today.
So thank you, Dr. Kane. It was such a wonderful session actually, because I remember last 3.5 decades, we have been only working on small molecules, trying to synthesize and trying to make different dosage forms and all that. But I had attended a conference on microbiome at Boston and the World Trade Center 2 years back, and really was very keen to know what are the different things in terms of gut-brain axis, and so many different things they are trying using gut microbiome and even microbiome on the skins. I think looking at all these technologies, and when I look at companies who are working on this, I don't see Pfizer or GSK, but not to that extent, but yes. But there are many small companies which are actually having intellectual capabilities and newer scientists and brains who are actually doing some disruptions in these technologies and then selling their whole company or the product to the big companies. I think there is a lot of scope in this, and thank you for bringing up to. I see that there are a lot of pharmacy students on our this session, and even to the academicians who are there. I think it's very important that the microbiology part of pharmacy should be strengthened further, so that these technologies, right from trying to practically implement them in the lab, and so that when they come to the industry, it becomes very easy for them to develop. Understand the development part, and as well as how to take it to the technology transfer and to the big manufacturing facilities, because the facilities also are very different than the formulation facilities. So thank you so much, Dr. Karne, for such a wonderful talk. Now I would like Dr. Alka Mukne to please take over.
And thank you, Dr. Ratnakar. And as Dr. Ratnakar has already done, Dr. Kani, thank you so much for that wonderful talk and tremendous and very valuable takeaways for young researchers. And as you yourself mentioned, a very futuristic talk, completely in sync with what has always been Dr. Tipnis's vision. And so the right thing to speak on this occasion, which is, and this forum, which is his memorial lecture series. So again, on behalf of the entire Endowment Trust, and all the participants, Bombay College of Pharmacy, a big, big thank you to you for taking time out and delivering this memorial lecture.
Thank you. My pleasure.
Thank you. And now for the last part of the evening, but the most important leg, that is the vote of thanks. And for this, I invite Dr. Reshma Tipnis to present the vote of thanks. Dr. Reshma Tipnis.
Good evening, everyone. I feel honored to propose a vote of thanks today on this special day. On behalf of my mother, Mrs. Tipnis, and myself, I would like to thank profoundly Dr. Anil Khan for accepting our invitation and spending his valuable time for us. Thank you so much, Dr. Khan, for delivering such a informative, very interesting, and really wonderful talk. And it's a great inspiration for all of us. And I know your association with Bombay College of Pharmacy and with my father was for many, many years. And hence, when you accepted our invitation, it really gave me immense pleasure. Thank you so much, sir.
Thank you.
I'm
Thank you.
extremely thankful to Mr. Nitin Maniar, Chairman and Governing Body, Bombay College of Pharmacy, Dr. Krishna Iyer, Principal in Charge, Bombay College of Pharmacy, Dr. N. Shivaprasad, the President, IPA, MSB, for their support. They have always supported us, given a lot of cooperation, and they have made all necessary arrangements required for today's session and for previous all lectures. And always they have given encouraging words and given guidance. Thank you so much. I'm very grateful to all of you. My heartfelt thanks to Dr. VM Kulkarni sir. He has taken a lot of efforts, and he has initiated for the initiative for the formation of the trust, and he worked really hard for the trust. Thank you so much, Dr. Kulkarni sir. Special thanks to Galvina Pereira ma'am and Mr. Sagar Patel for all this technical support. I will take this opportunity to thank all teaching and non-teaching staff of Bombay College of Pharmacy, current and especially those who were in service when my father was principal, for their huge support and cooperation, due to which it was much easier for the smooth functioning, and my father could do that. Thank you so much for all your support, and big thanks to all students of my father for their huge support, cooperation, lot of love and care given to my father, to my mother, and even to me. And especially even after the death of my father, all of them have continued to do the same. So I consider all of them as pillars of our lives, and big asset my father has left for me and for my mother forever. I really thank all of them from bottom of my heart. And thank you everyone for attending this session. Thanks all overseas people, USA, Canada, Shanghai. Thank you so much for attending and making this event a great success. Thank you so much.
Thank you, Dr. Reshma Tipnis, for the vote of thanks. And once again, thank you to all the participants for joining here. Now we have a lot of SIR students present from all over. If any of you would want to share your thoughts or want to speak on this occasion, we would be very happy to hear from you. So,
May I, Dr. Alka?
yeah, sure. Dr. Iyer, it is?
Yeah, it is.
Yeah, please.
Yeah, this is a message for Dr. Kane directly. Mr. Nitin Parayat send his regards. Nitin is my neighbor now. Talk about you. And in addition to that, I also complain for taking away a very good teacher, Vaishali Dikshit, to be closer to you. And I didn't realize it was your first friend. It's a pleasure, Dr. Kane, to have heard you today. And my regards
Thank
and
you.
respect to you and your family. Again,
Thank you.
regards
Thank
from
you.
Dr. Mr. Nitin Parayat.
Thank you. Thank you, Krishna.
So I think we also have Dr. Dhake here who would like to say something. Dr. Dhake.
Good evening to all. I'm very happy to be with all of you. I think it comes once a year that around 7 July we have the lecture, and generally I used to come to Bombay for that. I hope that we can start the lectures again where we all meet together. I was associated with Professor Tipnis for my PhD for 3 years, but I've been associated with the Tipnis family for the last nearly 20 years. I have a lot of respect for Tipnis madam and the love she has showered on all of us. I'm very, very grateful to the entire family, and thank you for this arrangement. Thank you very much.
Thank you, Dr.
Dhake. My mom has some connectivity issue, and so later half she is unable to join. Difficult for her to manage little technical things. Sorry.
So any of other students, sir students, Deepak, we can see Dr. Deepak Hegde. So if the admin can unmute him and get his video also on. Dr. Deepak Hegde.
Deepak is raising his hand.
Deepak has raised his hand. The admin has to. Yeah, Deepak, I think you can unmute and put your video on.
The settings are on for video and mic for all the participants.
Okay, good.
Okay,
Thank you.
can you hear me now?
Yes, we can.
Yes, Deepak.
Okay. Thanks, Dr. Kane, for an excellent talk. At least this year I had an opportunity again to listen to you, if not meet you in person. We've been in touch, except for this past 2 years due to COVID. We've not been able to meet, but it's an excellent talk from your side. And it's good that all of us had the pleasure of attending the talk. Thanks again.
Thank you.
To Reshma, Tipnis madam, for giving us this opportunity. And I look forward, Dr. Kane, to meeting you again in person when you are able to travel. Thank you.
Absolutely. Thank you.
Thank you, Deepak. Thank you, Dr. Deepak.
Thanks, Reshma.
Thank you, Deepak. So any of other sir students who are here who would like to share their thoughts or would want to say something?
This is Dr. Ashok. Can you hear me?
Yeah.
We can hear you.
Yeah, we can. We can.
Yeah, okay.
We can hear you. Can you put your video on?
Okay. I'm not dressed up properly, therefore I'm not putting my video on, but okay,
Ashok,
as you
Ashok
requested.
Ashok?
Yes, Ashok. Dr.
Okay,
Ashok.
can you see me?
Okay.
Yeah, okay.
Yeah, we can see you now.
So I think I understand everybody needs in-person meeting, this kind of event, but I'm a little bit happy today that because of this virtual event, I could attend it, so it's advantageous to me, and happy to see this kind of events are happening, and we should be organizing more and more such events in future. And thank you so much, and thank you, Dr. Kane, for your excellent talk.
Thank you. Thank you.
Thank you, Ashok.
Thank you, Dr. Ashok.
Thank you.
So any of the other attendees would want to share, sir's students or faculty or anyone else who would want to speak on this occasion?
Yeah, Dr. Mukne, can I?
Yeah, sure.
Yeah, it's very interesting that almost everyone has talked about all the beautiful things that are associated with Dr. Tipnis and his style of function. But the one thing which people missed out and which I put a lot of weightage to is that Dr. Tipnis also was a stickler for discipline. And I think a part of that discipline also has gone a long way in molding not only me as a student at Bombay College of Pharmacy and several postgraduate and PhD students. The drive to be very disciplined and very organized and have proper etiquette for different occasions is a hallmark of Dr. Tipnis. I'm thankful for that. Thank you, Dr. Thank you, Dr. Mittal.
Yes, as you rightly said, Dr. Iyer, he was very disciplined himself first. So the discipline part started with his own self first and then was extended to others. So yes, what you said is very right.
Thank you, Dr. Iyer. I think someone had raised the hand. Just
not able to see. Any of the other guests would like to share something about sir or
Hello, this is Dr. Mrinalini from Pune. Can I?
Yeah, sure, Mrinal. Hi.
Hello, everyone. I'm really delighted to be attending this Dr. Tipnis Memorial Lecture today. As Ashok rightly said, we would definitely want to attend such lectures in person. But due to this virtual event, we could attend all of us and practically meet. Thank you, Dr. Kane, for a wonderful lecture. And everyone here, it feels as if we are meeting and we are still a family that Sir built. I would like to specifically mention Sir's plus point, I would say, that though he was a principal with so many duties to perform apart from teaching in class, his speciality was that he would remember everyone by their first name. And today, as I have been teaching for last 21 years now, I know how difficult it is. I know that I can recognize my student after many years, but getting his first name right is really very tough. And sir could do that. And that adds a personal touch to his contacts. He was in contact with us for so long, and that's what I think has built our family. All of us, Dr. Tipnis sir students, we are, as we say, Guru, Bandhu and Bhagini. And with Reshma and Tipnis madam putting all of us together, I'm sure we will stay as a family in future also. Thank you for this event.
Definitely, Mrunal. Thank you so much.
So one incident
Can I speak?
share, which was actually out of context between me and sir. If Dr. Dhake remembers, he had organized some seminar at his college, and I and sir were invited. So sir came to my house, and then we went together. So it was in Nasik on the Sinnar road, and I was like, now I have to be with sir. So what to discuss? But you know what? From the moment we left, after some time, till I came to the toll naka, I think Bhoti, sir took me to Panipat, and he described the whole Panipat scenario. I think he had read the whole Panipat book, and he described the whole scenario, how and why we lost, or actually how we won. And then so I was really very impressed by, it's not only chemistry what he teaches, he knows so many things about history. And then when digging certain things, I found that in some I think New York Times or something in 1964, and I had also shared this, He had written in the Times that somebody says that Hyderabad is part of Pakistan, and against that author, he had really taken him left and right in the news, saying that if you don't know anything, you just shut your mouth. Hyderabad belongs to India and not Pakistan. And one thing was that he had also described in one of the article about how to make a Marathi dish called shira, and he had written it so nicely, that how do you make shira? And this was somewhere in 1964 or 63, at that time when maybe he was in the US, and that left a great impression on my mind that it's not only academics and administration, out of that, which is more of a family oriented thing. And that's how I realized that yes, he's a big family man, and that's why he has such a huge family.
Yeah.
Very good, Ratnakar. Thank you so much,
Very true.
Dr. Ratnakar.
Ratnakar, very nice. Ratnakar, I really like the way you were saying about the personal touch, what he said. As Krishna here also said, but before that, let me share that Kane sir, thank you very much. And it was a big interesting for a person like me to understand the research area of this. I was also expecting, since you talked about technology, you might also share how the technology has speeded up things or hastened up things. I would have loved to learn that also from you in the vaccine, in probiotics, in RNA gene therapy and all. But it was a wonderful session for me to get enlightened. Thank you very much. And I chipped in between. I was thinking why not to. But let me tell you honestly, the respect which started with Sir. Okay, we were in first year, and that was a time when Sir will be there in the corridor at one end at his office. The other end, if somebody is there, they will do an about turn, come down the stairs, and take another way. We never used to, until my third, fourth year, I am telling you, we never used to walk the path when he is coming from the front. Many students. But one day I saw that I was just about to take a turn, and he bent down. He's always in a white shirt, in a tie, with a tie pin. And with that clean thing, he's bending down and looking at something which is fallen down. He picked up those papers, and they were dirty papers. He picked it up and he kept it, went along, put it in the dustbin. Now, that is the day I started having a different persona about Dr. Tipnis. And since I was a student, then I could get a lot of personal things also, because that time we used to stay in the quarters behind, and we used to work at late night and early mornings also. We used to work 24 hours. I don't know whether today's students do that, but we used to work for 24 hours. So at night, 10 o'clock, in his pajamas and kurta, he will just come. Ashish, kai khalla kare. So he will be very much concerned whether you have had your food or not. And I remember twice that he has brought or sent that through our watchman, I don't remember his name, tea in the morning at 6 o'clock when he knows that we are still there. So he is disciplined, yes, no doubt about it, but he was at the same time trying to get personalized with every person and having a care and concern for every person in guidance with a genuine thinking. In fact, when he was with the pharmacy and my office was next door in Sun Pharma, he used to call me, and at that age also, in front of that tea tapri, we used to have a tea and have a good discussion also. So down to earth, no airs, and that's why he is respected. He contributed to many people's life, and that's what I see his biggest strength. Thank you for allowing me to speak that.
Thank you so much, Ashish.
Ashish, your talk, what you are talking is not audible.
Sorry, sir. I think I don't know that. Did anybody else had a problem?
I could hear it.
Okay, thanks.
Thank you.
I'll share with you, sir, afterwards.
Galvina, I hope you are able to hear it well because you are recording it.
Yes, sir. It was clearly audible. Yeah.
So, if there aren't any more people who are interested or any more guests who are interested in talking, then with everybody's permission, maybe we can close the session.
Yes, thank you, Dr. Alka.
Thank you so much, Dr. Alka.
Thank you, Reshma. Pleasure is all mine, always. So thank you so much, everybody. Special thanks, Dr. Kane. Thanks to everybody who's made time out and joined us here. This is the fifth. This was to be the fifth in the series of the memorial lecture series, Dr. Tipnis Memorial Lecture Series, and we will continue the good work ahead. With that promise, we disperse today, and stay safe, all of you. Please take care, and thank you so much again.
Thank you so much.
Stay
Stay
safe.
safe, stay healthy.
Thank you, Dr.
Thank
Kane. Thank you, Dr. Kane. Thanks, Swaraj.
Thank
I'll
you,
be in
everyone.
touch.
Thank you all.
Absolutely. My
Thank you,
pleasure.
Dr. Khan. Thank you.
Bye
Thank you.
bye.
Bye.